in a very interesting and latest trending topic, the network pharmacology and pharmacogenomics and approaches in the drug discovery. Because drug discovery is a very important process nowadays. The repurposing drugs as well as identifying the new drugs, overcome the various hurdles in the market. So the new innovations and invention is most welcome in the field, especially in the pharma. So that is the reason we have planned this FTP to fill the gap between the faculties, especially the academician as well as industrial collaborations. So we have planned here. So I warm welcome all the delegates. Now I request one of our colleague, Harinandini Madam, to give the intro about Prashant sir.
Hello. Sir,
Hi,
my
madam.
voice is audible, sir.
Audible,
Gre-
audible.
Greetings to one and all present here. Myself, Hari Nandini, Assistant Professor, Faculty of Pharmacy, BL. I take an immense pleasure in welcoming the speaker of the session, Dr. Prashanta, sir, working as an Assistant Professor in Reva University, Bioinformatics Department for the past 5 years. He did his PG in MSc Biochemistry from the Oxford College of Science, Bangalore, and PhD in Genomics from NTU, Singapore. He has published an enormous number of publications in both national and international journals like Journal of Biomedical and Health Informatics, Journal of Applied Biology and Biotechnology, etc. Dr. Prashanta Sir has also published in various conferences. Special mention to this publication on pharmacogenomics approach as personalized medicine for type 2 diabetes mellitus and differentially expressed neurogenin 3 gene in neuronal cell development of pancreatic cell. He has also organized countable successful workshops and FTP programs since 2017 on various thought-provoking topics. Now, I hand over the session to Dr. Prashanta sir. Thank you.
Thank you, ma'am, for the nice introduction.
Thank you, sir.
Shall we start the session now? So, good morning, all. I hope maybe you're all audible my voice. So, today my topic is mainly on network pharmacology and pharmacogenomic approaches in drug discovery. Maybe people are thinking that why the pharmacogenomics topics, because my main research area is mainly on pharmacogenomics, because a lot of research works what I'm continuing from past 12 years, and this is my 13th year in this particular domain, and I have published many research papers in pharmacogenomics also. Maybe in my CV, I have not included much of pharmacogenomic related, but most of the collaborations works are as like most of my research works are related to pharmacology. But coming up to main research here in this particular topic, to understand about pharmacogenomics. So because everybody thinking that why this, because we know that the genomics is separate and pharmacology is separate, but how it will be combined together with genomics and pharmacology, and what is the effect of that? So that we wanted to understand, and at the same time, like how these pharmacogenomic approaches will be current a trending domain. So later on in the slides, I'll just continue how it will be benefited, and how particularly like pharmacy people, they can able to use these pharmacogenomic techniques in their real life, so that they can able to develop a very good medicine for each individual diseases. And at the same time, I just wanted to discuss about the applications of pharmacogenomics also, then computational methods in pharmacogenomics. So what are all pharmacogenomic approaches we can able to use it, like the type of tools, databases, softwares. So how we can able to use it for understanding about pharmacogenomic approaches. And at the same time, we also include about network pharmacology, because no, in the topic I have mentioned about the network pharmacology and pharmacogenomics. But now I have included this network pharmacology at the end. So why this network pharmacology I have mentioned at the end, and how it will be related to our pharmacogenomics. So those kind of relations also I'll tell you. And at the same time, we also to understand about the applications of network pharmacology. So these are all the topics what I wanted to discuss today. And I hope maybe once you will, maybe if people are having any questions, so kindly raise your question so that I can able to answer immediately, continuing my presentation. So coming up to major topic about pharmacogenomics. So we wanted to understand about the reality of what exactly the pharmacogenomic is. Maybe I hope you have heard about what is pharmacogenomics and all. Maybe uh, in your current research and all, you have gone through with what is pharmacology or what is pharmacogenomic, right? Is it audible? Is it understanding?
Yes, audible, sir.
Yeah, sure. So here I just wanted to continue with the pharmacogenomics. So here, to know about the pharmacogenomic, because to understand about the variabilities existing among each individual patients, because now what we thought that every individual patient means because diseases are so many kind of diseases will be there, and each and every type of disease we know about the different type of drug molecule, right? And each drug molecule, while injecting to individual persons, because we know that one particular drug is given to multiple patients, how best this particular drug is more effective to that particular patient. Now, based on the body size, based on the age, based on the sex, or based on the hepatic characters, or based on the renal functions. So everything is influencing to the drug mechanism here. But if it will be influencing to the drug mechanism, how those drugs are really supporting to the patient. So that is a major concern for everybody. And at the same time, like because we know about the certain examples that one particular drug will be given to thousands of populations, whether all thousand populations they can able to accept the same drug. Right? So in this case, how best the efficacy of that particular drug to the patient, and how best the drug can able to control the disease. So these two kind of concepts people have to raise the questions and keep on asking that. See, suppose if I'm getting a one particular drug molecule, or if I'm taking one particular drug, how that drug is more effective to my particular body, because our body conditions are totally different, and we are all changing our environmental characters from one place to another. Like because some people are staying in the cold regions, some people are staying in the hot regions. So based on the environmental factors also, it will be influencing that how the body conditions can be supportive to the particular drug molecule. Right? So in these cases, there are a lot of challenges people are observed, and in many cases we know that some of the drugs can be supportive to certain populations, or some drugs will be supporting to another set of populations. But same type of drug, it's not going to support for all of the kind of populations, right? So in this case, to understand about it, okay, how the pharmacogenomic will be supporting to know about each and every drug molecule for each individual patient, right? So in that particular cases, so in the term itself, we can say about pharmacogenomic. Pharmaco, nothing but drug or pharmacological characters of chemical, and genomic, nothing but gene, right? So how this gene can be responsible to understand about the drug characters and behave, and to show their efficacy to the particular patient, and to control their disease conditions. So those kind of things to be understand. So for that sake, I just given a overview about pharmacogenomics. Here, to understand about the pharmacogenomics, first of all, we should know about how the genome-wide association studies will be supporting to understand about pharmacogenomics. Right, particularly about genetic factors are more influencing, because every individual persons they have their own genetical factors,
Maybe these genetical factors comes from the hereditary characters because of each individual population. Each individual populations vary their characters, right? Sometimes like genetical factors may supporting to that particular drug, or sometimes it's not going to support, or sometimes their metabolic activities are different, or sometimes maybe the drug can be accepted in their targeted region, but sometimes it will be rejected also. So there are so many factors which are influencing to the drug metabolism. At the same time, how best it will show their therapeutic targets. Because nowadays, actually, like you know about drug repurposing mechanism. The drug repurposing mechanism to understand about multi-targeted kind of drugs. Because so far, many drugs, whatever it will be produced in the industry, are single targeted or maybe one or two targets alone is there. But we don't know that whether this particular drug can be really is supporting to that particular disease or not. So with that cases, so people have to understand about how to identify a therapeutic targets of each individual patient, right? So right now, whatever the existed drug molecules are available, so that will be like a group-wise treatments, like a traditional method of treatments are there. But right now, whatever the research work we are focusing is completely based on the personalized kind of medicine. So each individual patient to be taken. So that particular patient to understand about how their genetic characters are available, what kind of genes are getting mutated, how the genes are getting mutated, and what kind of a drug will be influencing to involve in the metabolism or else to show their effectiveness with the targeted region. So all those kind of things to be understand. So people have to go with this type of a personalized treatment. So that's the people it's having a dietary substances. Because there are so many factors which are influencing, but in these cases, how those drug efficacy is presented and what is the toxicology or what is the toxicity ranges of that particular patient. Right. So with that cases, I have to know about pharmacogenomics. So people have to detail to study about how the genome-wide association studies will support to identify the type of genes and the type of mutations. With that regard, so I just wanted to go with in detail about the pharmacogenomics. So at the same time, people may also ask a question, like, what is the differences between pharmacogenetics and pharmacogenomics? Because here, genetics also is one of the part of genomics. But why particularly people they'll mention about pharmacogenomics, but why can't it pharmacogenetics, right? So in this case, to understand about the differences between pharmacogenomics and pharmacogenetics, here, because both are interchangeable, so we know that like a pharmacogenetics and pharmacogenomics both are interchangeable. But in the pharmacogenomics refers to a whole genome, like a complete person, like how the person, the genome is presented. Like we know about the Human Genome Project. So in the Human Genome Project, there's a total of around 3.2 billion base pairs what they have identified. But out of 3.2 billion base pairs with that, how many base pairs or how many genes are influencing to cause each functional character? Right? Maybe some genes are involved in the cancer, some genes are involved in diabetes, some genes are involved in some other diseases. So there are so many types of diseases, so many types of genes which are influencing to cause the disease. But in this case, how those genes are recommended or how those genes are influencing to involve in the disease condition, and at the same time how these genes are responding to that particular drug. So whatever the drug we are injected to that particular drug, how the genes are going to behave. So those kind of things also need to be understood. That is comes as a pharmacogenomics. So here it will be showing as a genomic techniques, transcriptomics techniques, metabolomic techniques, proteomics techniques, all are responsible to understand about pharmacogenomics. But at the same time, you also should know about pharmacogenetics also. Because pharmacogenetic is mainly focusing about the single gene. So here we have to focus only on one particular gene alone. And that particular gene, how it will be involved in various pathways, either in the metabolic pathways or else in the systems or else in the biological pathways. So how the single genes are involved in pharmacokinetics or pharmacodynamics, because once the drug is injected, how this particular gene is going to be altered, their functional characters, or else what kind of variations will be there in certain populations. Because pharmacogenetics will be studied about the type of population what we are going to be selected for the study, right? Maybe, for example, one set of population, maybe I am going to select some black people, or maybe I am going to select an Asian population, or if I am going to select an Indian population, or if I am going to select an African population. So, whatever the population we are going to take it, in that particular population, how that particular gene is going to be altered, the functional characters, right? So, those kind of studies we can able to say as a pharmacogenetic. It's a kind of a genome-wide association study. That's why we mentioned about a genome-wide association study. Maybe I think you're understanding about the concept, what I am explaining. Yes. So is it understanding? Are there any questions from anyone?
Yes, sir. It is understanding only, sir. Actually, we muted all the participants' mic.
Okay. Okay, sir. Sure. Because if
Yes.
because I'm continuously explaining and maybe people are can able to grasp my words or not, I don't know. That's why.
No, sir. It is audible only. The problem is whether unwantedly some noises is arising between the presentation. So we muted all the viewers' mic as well as. So when the questionnaire times, we open the mic and they can ask us.
Okay. Yeah. Sure. So yeah. I'll continue. So that's why here to know about the pharmacogenetics and pharmacogenomics. So pharmacogenetic is mainly focusing about the single gene influence with the different pathways that will be based on metabolic pathways or based on biological pathways, and at the same time how those pathways will be influencing to cause a pharmacokinetic and pharmacodynamic alterations. Okay. So that is a one way of analysis. We generally call it as a pharmacogenetic, but pharmacogenomic is totally different. So this will be for each individual population. It's not like the group of population. So each individual person also we can able to take like how the genome of that particular patient is there and how the genes are going to be responded, what kind of metabolites are presented with each individual patient, what kind of proteins are influencing to their disease condition. So those kind of things to be taken it with each individual population, and then to identify what kind of drug can able to respond to that particular patient. Okay. So those kind of analysis we generally go through with the pharmacogenomic. That's why. So here I'm given the differences, like one is in the pharmacogenetic, one is in the pharmacogenomic. Here, why this pharmacogenomics is necessary right now in this particular present condition? Because we know that there is a lot of research work, because everybody's focus is how to develop certain kind of drug, right? For whatever the disease comes, so to that particular disease, we have to identify certain molecules. That particular drug should control the disease. So that is only the aim for every individual researcher. Either it be related to on pharmacy or related to on chemistry or related to on biotechnology. But everybody's aim is to control the disease. But if they want to control the disease or if they want to cure the particular disease, so what kind of a method they can able to take it? Because I know that
the pharmacy people, they'll go with in vitro or in vivo kind of studies maximum of the time. But chemistry people, they'll focus more mainly into the synthesis and then talking studies kind of thing. Biology people, they will study about in vivo analysis or in vitro kind of analysis. They will go with the gene-based, protein-based analysis. So there are different type of research works are going on in this particular domain. But why this pharmacogenomics is necessary right now? Because this is a question for everybody's, right? So with that case, I just wanted to give a clarity, because this particular method is mainly focusing about the genes, right? So each individual patient, each individual person from morning to, because how the genes are going to be altered in their daily life from once if they'll go to bed, or after once they'll wake up, or till their life cycle from the day one, okay? After the person is born and whatever their complete life cycle is there, every fraction of second their genes are keep on altering, and these genes are going to be varied from each individual patients or each individual person. Maybe, for example, if I'll take one individual population or in one individual family, maybe for example father, mother, or brother, sister, like that within the family, if you are going to take also, whatever the genes you have, that same kind of genes is not present in your population, maybe in your family. Yes, I know that the human is having a 3.2 billion base pairs, and we know about around 30,000 genes are presented. That is, a functional genes are presented. Maybe around 18,000 proteins are presented. These are all, it's a common kind of thing that will be there for every individual population. But when it comes to particular conditions or particular expressions, so how these genes are going to respond? Okay, out of 30,000 genes, how many genes are going to be respond for each and every condition? For example, maybe if I'm getting hungry, or if I'm getting some anger, or if I'm getting some love. So with that case, how these genes will be respond to that particular activity? So those kind of things to be understand. So every individual population, these genes functions is going to be varied. Right? So with that case, the drug also, if you are going to be injected to that particular condition or to that particular patient, whether the drug is really supporting to the patient or not, we don't know. So for that sake, to understand about each and every concept, so we have to identify different type of drug targets. And at the same time, to understand about the drug transporters, and at the same time, to understand about the drug metabolizing enzymes also. Because we know about there are certain number of enzymes are presented, because we know in the pharmacokinetics while studying like admin properties like absorption, distribution, metabolism, excretion, toxicity, like that while studying, we know about certain kind of enzymes are involved in metabolic pathways or metabolic reactions. So those metabolites and all, whether what kind of gene can be responded or to activate to that particular enzyme to show the functional activities, right? So that complete type of studies, what we are going to take care here is in the pharmacogenomics. So if people they will understand about the pharmacogenomics, definitely they can able to identify different type of drug molecules. Okay, that's why I just given about the overview of pharmacogenomics, and here to know about the pharmacogenomic characters, mainly on genetic variations, and at the same time with that genetic variations like metabolizing enzymes with different genotypes, and also on transporter genotypes, receptor genotypes. So like this, all these things are there. But I know about each and every research, but nobody is focused about gene-based analysis. Nowadays, it's a current trend in India, not only in India, throughout the world. Pharmacogenomics is a current trend. But how best these pharmacogenomic drugs are, how best the researchers are using this particular technique to go for a research in medicine. So first, people have to understand understand about the pharmacogenomics, study so here to understand about the genetic variations and at the same time changes in pharmacokinetics, changes in pharmacodynamics, and also like what kind of inter-individual variabilities are presented with each populations that we have to understand, right? So that's why like here the pharmacogenomics is one of the biggest topic, and at the same time how to understand about the pharmacogenomic approaches and how to test those pharmacogenomics. So here I have given one simple example. For example, in the traditional prescription method of medicine, like one particular standard drug is there, so that same drug is going to be provided for thousands of population. But in these thousands of populations with that, how many populations are going to be means this dose works for most patients because they are going to give a certain uh, dosages for based on the age, based on the height, based on the weight, body weight. So they are going to recommend, yes, this particular dosage is recommended for this patient. But is that really that particular patient is accept that particular dosage or not? Right? So those kind of things and all to be understand. So definitely there is a limitations here while going with the traditional prescription methods. That's why now people are little bit uh, focused about prescription of drugs based on different categories. That's why like people are said about prescribed medicine, predictive medicine, or personalized medicine. So in this particular personalized medicine, so people have to take their particular genome to check with their variabilities. Based on the variability, what kind of drug they want to inject to that particular patient, they have to inject to that particular patient so that they can really get into better results. So that's what the pharmacogenomic approaches will be supporting. So this particular methods to know here, so we have to identify the type of SNPs, single nucleotide polymorphisms to be identified. So here SNPs that can change clinical outcomes. So why we have to identify the SNPs here is these SNPs sometimes it can change their clinical outcomes also. And SNPs that can change the drug responses, SNPs that can change pharmacokinetics, SNPs that can change activity in vitro, conservative amino acid changes, non-synonymous SNPs in exons, exon-based changes, and RNA-seq. Because in the earlier slides I just mentioned, like in the pharmacogenetics and pharmacogenomics while explaining, here we have to go with the genome-wide association study. So in this genome-wide association study, to understand how those genes or how those genetic mutations can alter each and every functional character. Maybe it can able to change their clinical outcomes, it can able to change their drug responses, or it can change the pharmacokinetics. But nobody is focused about, I know about from past 12 years what I am doing research in this domain, but maximum of the researchers, they are not focused about what is the mutation or what kind of gene changes, what kind of protein changes, what amino acid changes are there. So all these things and all, nobody is bothered. They will understand, yes, this is a protein is a target. So in this protein, yes, my drug will go and bind or not. If it binds, then they will say that, yes, you can take that particular method into an in vitro analysis or in vivo analysis and take it for further research. But how best that drug discovery is held? So all these things, we don't have a proper answer. That's why now people have to little bit focus about pharmacogenomic approaches. So for that, what kind of techniques are really required? Okay, if you want to go with pharmacogenomic studies, so first of all, we have to collect with each individual sample collections from each individual patient, because this is not like a the group of medicine. So I'm mainly focused focusing about the personalized medicine or predictive kind of medicine. So for that sake, to know about the predictive medicine for each individual patient, so you have to take their particular sample. Then to isolate a DNA, to go for a DNA amplification or PCR techniques to see that which gene is causing a disease, or else to go for a DNA sequencing. So here in the DNA sequencing again, I have a complete list of experimental procedures like this kind of thing. So we have to go for either in the genome-wide genotyping analysis, or else targeted genotyping analysis, or whole exome or whole genome sequencing analysis. So actually, like right now, so people are more preferred about whole exome sequencing for these kind of experiments, and particularly like a molecular diagnostic experiments and all. So many molecular diagnostic industries, they are focused about these kind of techniques with pharmacogenomic approaches. So mainly on genome-wide genotyping analysis, what they'll do are targeted genotyping analysis they'll do, or whole exome sequencing or whole genome sequencing experiments. So from these three experiments, so here we have to apply with computational prediction method. Actually, this is a very simple kind of experiment. Like only the cost effective is the sequencing alone. That too in India, it's a cost effective thing is whole exome sequencing. Maybe around 20,000 to 25,000 it may cost for each individual patient, each sample. But computational methods nowadays, like actually we have very good advanced softwares are there, tools are there, so that we can use those tools or softwares to analyze data with our own itself. And it's like if you just give, because there are a lot of online pipelines also it's available. Once the data is generated, sequencing data is generated, upload that particular data into a database or the tool, so that immediately you will get your results also. But this is one of the major techniques people they have to use it to know about each character. So after once it will be understand about the computational predictions, then people they can take with that particular result into an in vitro or in vivo functional analysis, or they can go with the different replicates of the studies also. So these kind of techniques usually they will prefer to go for genomic analysis. Other steps for designing of a clinical pharmacogenomics is that for hospital settings. So I just given here in the flow chart. Choose drug gene pairs or determine variant alleles, perform genotyping, report results. So usually like what I suggested here is like a computational methods and all. So people they can able to take a samples and isolate their DNA, amplify their gene, and then go for a sequencing. What they are going to report here is so what kind of a drug can able to pair to that particular gene, and to determine the type of variants, because these things and all already we have our own pipelines. So I also have I have developed certain pipelines for variant analysis and all. One example I show here. See what I have done with genome pharmacogenomic analysis purpose. This is a complete pipeline what I have developed, and this type of pipeline will support to identify the drug based on the personalized character. So I'll show you the some results here. So this is the data set what I have used, because the data sets are already existed in the databases. So that one only I have used. But here the sample what I have selected here is only related to a type 2 diabetes. See here I just mentioned about the type 2 diabetes, but the diabetes is in the multiple stages. I got the diabetic patients from 3 years of age to till 80 years of age group, but here I have selected only in the early stages alone, like 24 years to within 50 years of age group. Only I have selected for the analysis, but I have a data set of 196 data sets are available from for the patients of 4 years of 3 years baby to till 80 years of old person. So here is a data analysis what I have done. quality check, all those things, mapping. These things are quality analysis results. And this is a variant analysis. So I mentioned about pharmacogenomic study to know about what kind of a genetic variants are presented, so that need to be identified. So those kind of things I just done with the analysis. And here is a predictions. How many are non-synonymous are there? How many synonymous are there? So like this. So here I'm just recommending that what kind of genes are mutated and what are the SNPs we have identified with the patients. So here certain common genes what we have identified in each and every chromosome. So here I just mentioned about the chromosome, function of the chromosome, and reference allele, altered allele with the gene name, and what is the amino acid changes, reference amino acid and altered amino acid position in that gene, then SIFT score, then DBSNP ID. So SIFT predictions also we have done, and those RSIDs we have to use it for further our drug discovery purpose. While understanding about the complete statistics, so in the pharmacogenomics, in that particular tool, okay, whatever the pipeline I have shown in that particular pipeline, so here is the drugs what we have screened for that particular gene. So here you can see about the pharmacogenomic approaches in that. Here is the clinical annotation ID is given. Variants are haplotypes of gene. So here is a gene names we have mentioned. Here is evidences, level of evidence we have given. Like a level of evidences is 3. Maybe you're all known that level 3, 4, 1 to till 5 is there. So 1 is not recommended the drug. 2 is only concerned with the doctors. So like that, maybe the pharmacy people, they know about how the evidence is to be obtained. But here, all drugs are related on level 3. So it will be prescribed by the doctors. Level modifiers, here tier 1, like that we have mentioned, like a tier 1, tier 2, like that we have given. So tier 1 is highly recommended, tier 2 is moderately recommended, tier 3 is less recommended. Okay, so like that we have segregated the scores, and here is the score values are presented, and evidence counts we can say about some are efficacy or toxicity. So like this we have given the different predictions also. So here is a drug what we have recommended after the evaluation. So after once we done with the overall analysis, pharmacokinetic analysis or pharmacogenomic analysis, and finally these are the drugs what we are recommending to these particular gene. But right now we don't know. For example, metformin is given as a drug for the diabetic patient, but whether the metformin, which one is the target, nobody knows that, right? So we know that yes, the metformin is taken, the blood sugar level is going to be controlled. Only that particular character alone we know. Or if anybody will say that if the metformin, some people they will say that yes, it will be one of the ion signal transduction inhibition. Only certain characters alone, people they can able to give their but here we have clearly mentioned that what is a drug and what is the target, means targeted gene, and what kind of mutation positions that gene can able to support or the drug will go under show their effectiveness. So here is the filters also we mentioned. So in this particular gene mutation, so this drug will go and bind to this particular gene. So those kind of predictions we can able to make it by using a pharmacogenomic approaches. So like this and all we have mentioned. And then we went with a complete biological pathway analysis also. So in that biological pathways, whatever the genes we have given here, so because we wanted to confirm, yes, these genes are really there in the diabetic condition, or what kind of pathways these genes are influencing, and what kind of a cellular region that gene is functioning. So that we wanted to understand. So for that, we went to a little bit of more analysis. So here we came to know that certain means whatever the genes we identified, so those genes are presented here in the beta cell development, or regulation of gene expressions in beta cells, or insulin receptor recycling, or diabetes pathways, or glucose metabolism. So here is a gene list. Right? So these type of genes are presented in this cell development. And the drug also, whatever the drug we are recommended, so those genes also will be presented in this particular region. So those kind of analysis we can able to perform by using a pharmacogenomic. So here also is another one more diagrammatic representation I give you similar to this. So till that is a pharmacogenomic. So I just wanted to continue as a network pharmacology also. Because here I wanted to link between network pharmacology and pharmacogenomic. So that particular part I'll take it later. So one part I explained about pharmacogenomic. Now I'll go with the network pharmacology. Later I'll show together like how this network pharmacology is supporting understand better way to understand about pharmacogenomic. So now I just wanted to go with the network pharmacology. So in this network pharmacology is one of the interdisciplinary field that combines the systems biology and pharmacology to understand the complex interactions between drugs, targets, and biological networks. So to understand about the interactions, because we have to know that how the genes means how the drugs are interacting, how the targets are interacted, and what kind of biological networks are presented. This approach moves away from the traditional or one drug, one target to a holistic view to recognize that drugs often interact with multiple targets also. But nobody is focused about multiple targeted drugs. But nowadays it's a trend that yes, earlier only one drug, one target is identified, but now people are going with a multi-targeted drug analysis also. But how to identify the multi-targeted drug and what are all the concepts we can able to use it here to understand about multi-targeted characters. So for that sake, to understand about network pharmacology, so we have to go in detail about key concepts. That is a polypharmacology. So we can mention about because no, the overall term, whatever we mentioned about the network pharmacology, or else I can say about polypharmacology. So in this polypharmacology, so there are different steps we are going to use it. There are 5 different steps are there. One is the polypharmacology, biological network, systems biology integrations, drug interactions, and personalized medicine. So here in the polypharmacology, so first, apart from going with a single targeted drug, we have to go for multiple targets. So if you want to identify a multiple targets for one drug, multi-targets. But how to identify those multi-targets, we don't know, right? So if you want to know about that multi-target, again, you should understand here what is the type of, means what kind of genes or what kind of proteins are presented in that particular disease condition. That we have to understand, right? That's why here drugs can interact with multiple molecular targets, which can lead to synergistic or antagonistic effects. But nobody knows that because we know about, for example, metformin is going to take, people will say that it will go under control our glucose level. So you know that what kind of a gene or what kind of a proteins will go and bind, means this drug will go and bind, and whether each and every drug is having only single gene alone is a target or multi-gene is a target. So how to know about these characters, right? So those kind of things to be identified, we have to go for multi-target identifications, and at the same time, drug repositioning also. So this drug repositioning, again, it's one of the biggest topic in this domain. That too particularly when people are going with the generic medicine purpose. So in the generic medicine identification, so people have to identify, because we know about one drug and one target concept, but only one drug and one target concept. If people they will go again, they have to spend a lot of money to synthesize the molecules, or if they want to produce a chemical, right? So for that sake, people they will go with the drug repositioning, so that they can able to identify the new therapeutic targets also, so that it can able to influence to other diseases also. For example, in the corona time, we got means immediately the corona disease came, and we don't know like what kind of a drug to be taken it for the treatment. But luckily, people suddenly they identified about a certain multi-targeted drugs to control the disease. Maybe an HIV drug, they started giving the injection at the beginning to control the disease. But whatever the drug is given, that is related to the HIV. But they are also given the treatment for corona also. So like that, some of the drugs is having a multi-targeted characters. But those characters and all, nobody knows right now. Right? So for that sake, people have to take an existed drug molecule to find out whatever the drug is presented right now in the market, take that particular drug to find out what other new therapeutic targets are presented for this particular drug, existed drug. So those kind of analysis to be performed, that is in the polypharmacology. That's why we particularly mentioned about polypharmacology or as multi-targeted analysis. So for that, if you want to go with multi-targeted drug analysis, you should understand about the biological networks also. So in this biological networks analysis, you need complex interactions to know about how these complex interactions are presented with the genes, how it will be presented with the proteins, and how it will be presented with the different metabolites. Okay. So these kind of networks to be identified, and how the interactions are presented, and how to understand about these effects in the biological system. So those complete analysis to be known. So people have to really know about their biological system. So how the networks are presented. Where is the node? Where is the edge? So how the nodes are connected, how the edges are connected. So that you can really develop a proper drug to that particular disease. And at the same time, go with the pathway analysis also. And then we have to integrate with the systems biology integrations, because without the omics studies, it's not possible. That's why I interlinked with the pharmacogenomics here. So here, omic data is more important, like a genomic, proteomic, metabolomics. So every omic technique is more influencing to develop a pharmacology. And at the same time, we also need a computational modeling methods also. Computational modeling methods, nothing but maybe an homology modeling or molecular docking or molecular simulation methods. So these things also, it's more important here to develop network pharmacology. And at the same time, to know about further analysis, so we have to go with the drug-drug interactions, that is combination therapies. So here, two methods are there in the drug-drug interactions. One is combination-based therapies, and another one is adverse effects. So in combination therapies, network pharmacology helps design combination therapies that target multi-compounds of disease networks and potentially increase efficacy and reducing side effects. Because the network pharmacology or drug repurposing main aim itself, so increase the efficacy and reduce the side effects, right? So that is only the major aim of network pharmacology. But at the same time, we have to understand about how different drugs interact with the within biological networks. So that also need to be noted here. Okay. So for that sake, so all these 5 points, whatever I mentioned, like another one is personalized medicine, that is patient specific models. So we have to take an individual individual populations, because once if you go for a drug repurposing, you have to check with the individual patients, like what kind of patients this drug can be supported. Because some of the drugs, whatever it will produce in the pharmaceutical industries, is not supporting only to our population. It also to support on certain kind of populations also. So here you have to specify that what individual patient or what kind of a population this drug can be used. Because our environmental conditions are different, our genetic characters are different, okay? Our genomic content is different. So that's why here we have to combine together with pharmacogenomic approaches along with the genetical approaches. So here is some of the applications of network pharmacology. Here we have to go for drug discovery and development methods. Again, target identification, lead optimization methods also we have to. And at the same time, to understand about disease mechanisms. So in the disease mechanism, pathway disruptions they have to go through. So different like biological pathways and biomarker discoveries and precision medicines through Ervelica integrated. So before going for the applications, so I'll just give overview like how the network pharmacology will support. Biology to network pharmacology, how it will be supported. So for that, I'll just give the overview here. One thing is to understand about the type of module and betweenness. So whether the number of shortest paths that go through a given node. Module is a group of nodes that act in the concept of to perform a specific functions, and hub genes to be identified. Hub gene a node with high degrees, and then degree the number of edges that is connected to a node, because these things we have to understand. That is a hub gene to be identified. Then which one is having a shortest path to minimize the path between any two nodes in a network. So these things are more important for us. So whatever the nodes are there, yeah, whatever this dark points is mentioned, that is a node, and whatever the edge positions is mentioned, like the type of lines where it will going to be end, that is in the edges. So those kind of things to be I understand. So to continuation of that particular network biology to network pharmacology. So what are all the things to be studied here is protein-protein interaction networks to be studied, and then gene regulatory networks to be studied, and metabolic networks also to be studied. So maybe I know that most of the people, they don't know about how to understand about protein-protein interactions. So everybody is not familiar with biology. At least whoever the researchers will be focused about network pharmacology, try to understand about how the protein-protein interactions are performed. And at the same time, gene regulatory networks also, because in the cell, we have gene regulatory networks, protein regulatory networks, metabolic networks, and drug regulatory networks, like receptor regulatory networks or receptor functions. So like that, there are so many types of regulatory functions which are presented in the cell. But once the drug is injected to the patient, injected to the cell, how these networks will be interlinked to control the disease condition, or else it will show their efficacy in that particular cell. So for that sake, here some of the things to be understand about how the nodes are presented, how the node interactions are presented, and how the up genes are there, where the cluster of genes are presented, where it will be connected, where it will be interconnected, how the nodes are connected together. Okay, whether the nodes are there in the sequential order or only in particular region alone the genes are present. So like that we have to understand. So that's why the network pharmacology is one of the biggest domain now. And at the same time, we can able to use so many different ways about network pharmacology. So yeah, I just taken one simple example about the traditional medicine. Because this traditional medicine, we know about right now anyway, people know about the chemotherapeutic drugs or whatever the synthetic drugs and all. But at the same time, we also to take care of the traditional medicine to be converting into a conventional, sorry, very good. So in this case, so we wanted to highlight about traditional medicine. That is a complex formulations we have to make it. That is a traditional medicine often used as a complex mixture of natural products, which contains multiple bioactive compounds. Because nobody knows that how to segregate proper. Because what my experiences I observed, like everybody they'll go for yes, I can extract one particular plant, I can take that as per particular disease. But is that complex mixtures what they are going to prepare, whether they are going with any kind of formulations, or if they are going with the formulation means whether they have tested with each individual drug. Because I know that people they'll go with the crude extracts, but very less number of populations only they'll focus about individual compound extraction, or else some complex mixtures. So that if they know with the complex mixture preparation, so that they can prepare a proper drug formulations here. So those kind of things to be understand here. So with that, people have to go a little bit of a combination of approaches. So in that case, I am giving here some of the overview of network pharmacology things. First thing is to know about the medicinal plants, and then we have to go with the data mining approaches. In this data mining approaches, we have to learn about different databases and different literature searches, and at the same time, laboratory experiments are required. And once if they understand about the data mining approaches, so I'll show you on different databases also what kind of databases we can use it for biological, this network pharmacology purpose. And here to identify about disease compounds and their targeted genes. So you can see here in the conditions what I have mentioned. Here is a disease and here is a plant. So here also to identify about the targets acting on disease, and in this case, the active compounds of plants to be identified. And here there are different methods what we have given. One is compounds target pathway network analysis, compounds with the gene networks analysis, then KEGG analysis and GEO analysis. That is a gene ontology predictions. I hope maybe you know about what is gene ontology. So anyway, later I'll tell. So after once people they will understand about all these characters, then we have to go for validation of those drugs with the specific targets. So with that case, people they can prefer about in vitro analysis, computational based in silico, in vitro, and in vivo analysis. So here is some of the databases what we can use it for. Medicinal plant characters to be analyzed. So DCMSP, then HERB, TCMB, MBank, HIT. So like these many different databases what we have right now. And some of the protein-protein interaction purpose, we have string databases there, UniProt databases there. Then disease related, we have drug bank, OMIM database, DisGenet, TTD, that is a therapeutic targeted drug. So like the database, these many databases, and the functional annotation purpose, we have David functional annotation database is there. Metascape is one of the genome annotation database, and at the same time, KEGG database also is a pathway database, or biological pathway analysis database. So by using all these databases, and at the same time, these are all it's a computational work. It's not purely wet lab work. So whatever the experiments or whatever the procedures are mentioned here, only that thing is here to identify about the phytochemical, and here is completely into a means a computational based method. Whatever the compound is identified, so that particular compound you can also search by using computational based method to understand about the targeted pathways or else like a gene networks, egg analysis, geo analysis. All these things you can do it in a computational based method or in silico based method. So by using those things, we have to predict a network. So in this particular networks, you can see about that there are so many targeted genes are presented. Every targeted gene. So here, if I'll focus about CNR1 is a main target because this is showing on a multiple networks here. And at the same time, there is a dark color also because we are applying some statistics here. So to know about which targeted region can we use it as a drug target. So here CNR1 I can able to use it as a target for these many number of genes. Okay. Maybe if any gene mutations are presented in these genes, if I give a drug to this particular gene, it can able to go and control these many number of gene functions. Okay. So like that is in the overall. Here is a completely computational based method. So the same like what I have mentioned here also, the same protocol I given here, but here little bit of elaborating kind of thing. And some of the practical steps in integration with the biological network pharmacology is data collections, network reconstructions, then compound modeling and experimental validations. So the major steps here. And here is some of the common targets, means whatever network pharmacology based people have identified. The growth factor related, these many number of targets what they are identified. Tyrosine kinase related, these many number of targets what they are identified. And a serine threonine protein kinase related, like certain proteins, GTP binding protein related, nuclear protein related. So common targets. These are all it's a common molecular targets for any disease, particularly on a tumors related. Okay, like cancer, whatever, like lung cancer, all that. So after once if people they will do with the network pharmacology analysis, here we have to understand about how the network to be built so that we can understand yes which drug we can able to recommend for what kind of functions, maybe related to anti-inflammatory, antioxidant properties, anti-inflammatory ones, like that each and every character to be analyzed. So we have to give certain coloring things also. And here we mentioned about whatever the triangle shapes are there. So these triangles and all it's a drug characters, and the circle shapes and all it's a genes. So here I have mentioned about the thing. So here the drug target interactions are shown as connecting lines between drugs, that is a compounds, that is mentioned as a triangle, and targets is mentioned as a circles. And the black nodes, particularly here, the black nodes represents that there is a targeted by ARBs on the formulations. Actually, these are all let's say experimental procedures. So we have to use the experiments. So here is a experimental thing how the network pharmacology we have to build, particularly about in silico methods. Mainly on sequence analysis, gene expression based analysis, protein-protein interactions, homology modeling, protein structure reconfiguration, then active site identification, assay developments. So like that, everything it will comes as a in silico method. And here also, this also an in silico based method, that is a lead identification and validation. Maybe you know about the overall drug discovery protocol, like disease target identification, lead optimization. So like that, there are many steps are there. But here I given only about in silico based method. And here is the database where the disease can be selected. So after once if you're done with the network pharmacology, those particular genes you have to select the protein and then you can identify there are mutations also, and then you can do a blast analysis. So the blast you can have different templates. To that particular template, you can do an homology modeling to check with the Ramachandran plot also, and also to check with the RMST values also to that particular target. And then you can see about how that particular target. So here, this is my targeted region. So in this target, if I prepare a drug to identify what the active sites of that particular target, and then to do an ligand-based or structure-based docking studies. So like this, we can see. So finally, after observing about the overall thing, so based on the docking results, you can screen those molecules, and then you can take those molecules into an in vitro and in vivo studies. So like this, we have to take care of overall network pharmacology. That's why here we can interlink whatever the genes we are identified from the beginning, like whatever the targets we identified based on the gene. So those genes we can take it and we can interlink with network pharmacology to understand about the gene-gene interactions or gene-protein interactions like this. Like this, we have to interlink. So based on this, whatever the targeted gene is presented, to that particular targeted gene, we have to take drug, and then we can find out the docking. So this is how usually network pharmacology is working. So I hope maybe you understand my presentation. So any questions?
Thank you, sir, for your wonderful presentation. Actually, you have given the elaborative idea to the bioinformatics tool, how we can utilize in a pharmacy and how can it is used in the in silico analysis as well as pharmacological network systems, as well as to identify the in silico target identification, lead validation process, or really it's a very nice presentation, sir. Even including the database to identify the tool and various ways, practical approach. It's looking like a workshop on how to utilize the process. Really, I thank the amazing team to choose us a resource person. I request Vasant to unmute the participants. Any questionnaires from the participants?
Yes, sir. They can raise the hand. They can ask questions.
Yeah. Anybody there having the doubts, they can raise your hands. You can freely ask your questions. Any questions? Sir, how it is suitable to identify the disease based on the gene modification? Is there any practical approach than any commercial process in India, sir?
Yes, sir. Actually, right now you can see, sir, most of molecular diagnostic laboratories, what they are working is to understand about the disease characters. Because now many type of diseases, without knowing us, people are getting died. For example, heart attacks are there. Just an example, or else like a cancer also is there. So maybe the diseases at the beginning, people will be neglected, right? And if those neglected kind of characters and all, so one day it will show sudden death, right? So if the sudden death is there, then definitely why people they could not able to understand about their body condition at the beginning. Like for example, I have done my own genome sequencing. Me, my complete family, I have done my sequencing. And then I done with a complete analysis, and I have preserved my data now. I have preserved my data. I know about my family profile and all. Another 10 years, I can definitely assure that yes, in my family, I won't get any kind of disease characters. Okay. So like that, these kind of genomic techniques are more influencing to understand about the family conditions, right? Because no, every individual populations, at least maybe you know, in maybe for example your family, maybe your father or mother, maybe your grandparents, somebody is having some disease, maybe cancer or something. But we think that yes, I'm healthy right now. So that is our feeling, right? Maybe if we will go for walk or if we will do a regular exercises, people will think that yeah, I'm fit, I'm perfectly all right. But we don't know what kind of gene modifications is there in our cell, right? So people, if they'll focus about genomic techniques to check with at least once in their lifetime, at least 10 years once, if they'll go for sequencing, they can able to take proper precautions also. But at the same time, so if people, they'll understand that yes, this type of gene is getting mutated in my body, immediately people, they can able to start preparing, means start taking the drug to that particular gene. But while taking a drug also, again, you have to evaluate the drug, whether the drug is really suitable to my target or not. So these kind
How
of
much
things,
they will?
yes, sir.
Yes, yes, yes. Still continue, sir.
So those kind of things to be evaluate right now, people are more focusing about pharmacokinetic approaches, like drug efficacy of each individual gene character. So that kind of research is going on in India right now, but it will take time. But in India, I think maybe another 115, 20 years. Still, it will take time in India to develop these kind of technology. But right now, I know about because I'm working from past 12 years about this same research, particularly on diabetes and cancer. Cancer related means only on colorectal cancer I'm working. But apart from colorectal cancer, diabetic things are more now. I'm working more on diabetic. But nowadays, people are little bit aware about. Yes, people have to go for pharmacogenomic studies. So that I think recently also you may know about reverse diabetic. We can reverse the diabetes, right? So we can reverse the diabetes means people are developing certain applications and they'll just put that particular chip to their hand so that they can understand about their glucose level regularly. But at the same time, here we are detecting certain gene, which one is mutating in their cell, what gene is mutating in their body, so that we can immediately take a precaution and we can sustain with that disease.
Yes, yes. So how much they will charge for one genomic? Suppose I want to take my DNA analysis, genomics isolation. How much they will charge, or any suitable diagnostic areas is available?
Sir,
Whether it is affordable to the common person?
right now, actually, it will be more affordable to anyone because earlier, you know about the genome cost, it will be like a lakhs together was there. But now it will be like a thousands. Hardly like, say, I have done my whole genome sequencing, whole exome sequencing, only for 22,000 rupees. The 2 years back. Okay. So now, for example, now if the person wants to go for whole genome sequencing or whole exome sequencing, hardly they have to spend on 12,000 to 15,000. 12 to 15,000 if they'll spend. So they will get their genome data also and their complete profile also they can understand. They won't get, means they can understand, yes, in my body, is there any disease in the future? Maybe we don't know, heart attack or maybe diabetes, maybe in a cancer, or maybe some other nervous diseases, or maybe Alzheimer's or Parkinson's. We don't know anything, right? So those complete list we can able to understand in the beginning itself. Actually, we understand in the earlier cases, one patient, he went to so many hospitals for treatment. But doctors, they done with the scanning, all those things, blood tests they did, and so many tests and all they did. Finally, they could not find what disease they have. But she's continuously unconscious. And finally we suggested them that to go for you can try with the genomic method also, see that how best results you get. So she went to the genomic technique. Finally, they detected that she had some problem in the brain, nervous cell. One particular gene is mutating, keep on mutating in the brain cell. So for that sake, she is getting unconscious in the regular. But in through scannings, MRI, they did so many protocols they did, but they could not find it. So those kind of unknown diseases also we can able to predict by using this genomic method. And commonly, like anybody, they can able to afford this particular technology now.
It's very nice and nice presentation. Any questions?
Any
Vasant
questions?
sir, anybody? Even you check the YouTube live, anybody there ask any questions?
No, sir. I asked to raise the hand, which is given in the options. If they are say, then I can share to you people. Sir.
Okay, I will
Anyone?
share the SAT details in the WhatsApp group. Anybody they are interested, anybody they need to clarify their doubts, you can make a mail to sir and you can clear your doubts. This is the time we close the session. Thank you for your wonderful presentation, Prashant sir.
Thank you for the opportunity, sir. Thank you so much.
Thank you very much. Now I request Harinendri madam to give the closing session speech.
Hello. Vote of thanks. This FTP has enlightened our knowledge with various new aspects. It's time to convey our heartfelt gratitude to the speaker, Dr. Prashanta sir, for sharing its valuable thoughts with all of us. Special thanks to our dean, Dr. R. Srinivasan sir, and HOD Dr. R. Saravanan sir, and management for their constant support. And I would like to extend my gratitude to all the faculty members for their tremendous support for successful completion of the session. Thank you, sir.
Thank you very much. I also personally thank all the persons behind in this. Each and every faculty they were behind in this to make the good successful in the FDP. I once again thank Dr. Prashanta sir and Radhaganavadi sir, Dr. Srinivasan sir, as well as our management and Vasant Kumar sir, all other members, those behind in this work, sir. Thank you very much, sir. We will meet next day one more speaker, Mr. K. Halim Shah from the Drug Safety Pharmacovigilance from TCS Mumbai. Thank you very much, sir. Thank you, sir. We can close the session.
ரெக்கார்ட் ஆயிருக்கு ரெக்கார்ட் எதுல சேவ் ஆகுது
செக் பண்ண கிளம்புறேன் கிளாஸ் இருக்கு இதில் என்ன பண்ணுறேன் உன்னோட மேல் ஐடியா சார் மேல் இருக்குது இது அவங்க இருக்கு இப்போ சார் வந்து லைவ் ஸ்ட்ரீமில் என்ன கொஷின் கேட்டிருந்தாரு லைவ் ஸ்ட்ரீமே ஓடல ஓடுதுமா
ஸ்டாப்
பாட்டு பண்ண